everyone, and welcome to my channel. This is the seventh episode of the Meet the Pro series, a series where I will be interviewing people in the field of biology and genetics to help high school students interested in biology. In today's episode, I will be interviewing Dr. Fred Marshall Winston. He is the John Emery Andrews Professor of Genetics at the Harvard Medical School, studying the effect of gene regulation in protein transcription, and I'm delighted to have him with me here today. Thank you for being here, Dr. Winston.
You're welcome.
So, I just want to start with kind of like a background question. How did you first become interested in biology and research?
I became interested in biology when I was in college at the University of Chicago. Um, Starting college, I had no interest in biology whatsoever, but I went to a school where everybody, regardless of major, was required to take a biology course. I tried to talk my way out of it and I failed, and they forced me to take biology. And I found it so fascinating. And the professor was such a good teacher that that first quarter of the year, I changed my major to biology
Oh,
and then joined the lab of the professor who was the teacher.
hmm. What was your major before choosing it to biology?
Math.
Okay, so you went from math to biology, and you never expected that.
Never. I was very surprised.
Hmm. Um, so right now, why have you chosen to work at a college, and why not rather work in the industry?
Well, first of all, I work at a medical school, not a college, but I work at a
Yeah.
university. Okay. Um, I wanted to work at a university because I wanted to be able to have students in my laboratory and to teach students in the classroom.
Okay. What does your current research look like?
Current research focuses on trying to understand what regulates transcription in eukaryotes, as well as chromatin structure. Transcription occurs on a chromatin template. We use the model organism yeast, Baker's yeast, the same kind you buy in the grocery store, which has been a fantastic model organism.
So now I want to switch over to some science related questions. I was reading your bio on the Harvard website, and I was wondering, why do you say that the function of most transcription is poorly understood? Like, could you kind of elaborate on that?
I said the function of most transcription is poorly understood.
Yeah, that was one of the things I read on there.
Oh, well, let me just say that let me revise what I said then, that
Okay.
many mechanisms for understanding how transcription is controlled is poorly understood. If we think about transcription in human cells, for example,
Mhm.
there's the transcription that goes across protein coding genes that is translated to make proteins. That's understood at some level, the purpose of that transcription, but there's also non-coding transcription that occurs across vast extents of DNA that does not encode proteins. And those are called long non-coding RNAs. And
Yeah.
the function of that transcription is poorly understood. In the
Okay.
yeast genome, which is much smaller, there's not as much space. There's still non-coding transcription. And again, for most of that, it's not understood what its purpose is.
But why would transcription like occur in those um intergenic or non-coding regions?
Oh, there are many possible reasons why that might happen. One reason is that the RNA itself serves as a product rather than encoding a protein. There
Hmm.
are some complexes, such as a ribosome. A ribosome is mostly RNA with some
Yeah.
protein stuck on it.
Okay.
There are other enzymes that are RNAs. RNAs can have structural roles. RNAs can be parts of protein complexes. There are probably many other undiscovered roles for RNAs. In addition, the act of transcription, rather than the transcript itself, can modify chromatin, can open it up, can modify the histone proteins that are part of chromatin, and that can affect the behavior of how the chromosome acts or what it interacts with.
Okay. Um, is the arrangement of the chromatin, is that involved in any way with gene regulation?
Yes, the arrangement of chromatin is intimately involved with gene regulation. Chromatin, if we think about chromatin as a nucleosome, so that's
Mhm.
150 base pairs of DNA wrapped around 8 histone proteins.
Okay.
In that configuration, other proteins are usually unable to bind to DNA. So in order for gene regulation to happen, often nucleosomes are in the way and other regulatory factors have to do something to knock them out of the way, either modify their structure, modify the histone proteins, or kick the histone proteins off of the DNA. So they are really very intimately involved.
Do they need to like unravel that structure, like the way that the DNA is wound around the histones?
It's believed that there is certainly some unraveling involved.
Mm,
Biochemical experiments have shown that when transcription occurs or some factors act on nucleosomes, that the DNA becomes more accessible to other proteins. So yes, I think unraveling, yes, it's probably not as literally unraveling like we think
yeah.
about it, but some loosening and then retightening probably happens during many processes.
Okay. And I was learning about CRISPR the other day, and I was like seeing how it's not always the most efficient tool when it comes to editing genomes. Like it has a lot of, I guess, um, drawbacks. So will we ever be able to like reliably edit our genome, especially like at a therapeutics level where if we use that tool, we can cure genetic diseases?
I don't think we can answer that today. I
Mm,
think that there's many, many more studies that need to be done. Certainly, a lot of people are thinking about it and debating it. But even when we use CRISPR on yeast, we see unwanted mutations arising that we didn't plan using the CRISPR tools that we have. And that's certainly a big concern. So I think it's too early to say. I think a lot of people, a lot of smart people are thinking about it, and we just have to wait and see.
Uh, do you have any opinions on CRISPR, like specifically around its sort of ethics?
Well, I think anytime we are thinking about tampering with the human genome in a directed way, there are serious ethical questions that have to be discussed, whether it's CRISPR or anything else.
Hmm.
So that's really all I want to say. I think one has to be extremely careful about any kind of modification. That's one reason I like working on yeast. We use CRISPR all the time. We're quite
Yeah.
happy to modify the yeast genome
Uh-huh.
however
Yeah.
we desire
Yeah.
and look at the consequences, and the yeast have not complained to us yet.
Okay. So finally, I want to end with a sort of fun question. If you could choose any extinct species to bring back, which one would you bring back and why?
Any extinct species? Oh my gosh. Which one would I bring back and why? Um, would probably want to, I don't know a specific one. I would
Hmm.
probably want to look at some recently extinct species of bird that was considered to be beautiful or
Okay.
everybody misses and bring that back or some like that.
Okay, that's really cool. Uh, all right, anyways, I think that's all we have time for today, though. Thank you, Dr. Winston. I
You're
really
very
appreciate it.
welcome.
Have
Bye
a good
bye.
day. Bye bye.
You too.